So Michael and good morning everyone. So today we are going to discuss about structure of enzymes. Okay, now structure of enzymes. Okay, as you can see from here, right, this is an example of an enzyme. Okay, and this is a closer look on the enzyme, and this is about the side chains of the enzyme. Okay, now without any further ado, let's start our discussion on the structure of enzyme. Okay, so enzymes, they are globular proteins. Okay, they are globular proteins that act as catalysts that speeds up metabolic reaction. And enzymes are globular proteins with precise 3D shapes that is formed, right, with primary, secondary, tertiary, and quaternary structure, okay, of amino acids. Enzymes, they are soluble in water because of the presence of hydrophilic side chains on the outside of the enzyme molecule. This is an enzyme molecule, right? Betul kan? Now, on the outside of the enzyme, there are many hydrophilic side chain. You can see here, right? Many hydrophilic side chain, and this hydrophilic side chain, which is on the outside of the enzyme, gives the enzyme the properties, solubility in water properties. Okay. Now, if you notice, the hydrophobic side chain, however, they are on the inside of the enzyme. Okay, yes, and that's very obvious because hydrophobic, apparently this gives the properties of fearing the water or insoluble in water. So this hydrophobic side chain tends to group insides of the enzyme precise 3D molecule, right? Hydrophilic side chain pula di luar. Now, enzymes only form complex with their substrates. Okay, means the action of enzyme is specific to their substrates. So normally enzymes are named for their substrates, and usually this naming of enzyme end in ase. Example, lipase. I think you can guess lipase is an enzyme who's involved with hydrolyzing with their substrate, which is lipids. Okay, protease involved. Protease is an enzyme that can hydrolyze protein. Betul kan? Okay. Now, enzyme has one or more active sites. This is an active site of this enzyme. If you look here, and these are active sites. So one enzyme, okay, can have one or more active sites. So active site is a cleft or pocket. You can see the cleft here, pocket here, okay, that is found on the surface of enzyme. Most enzyme, right? Most enzyme has active sites, right, on their surface, the surface of the enzyme. Okay, but some enzyme has active sites, right, inside their grooves. Meaning to say, in one enzyme, right, it has grooves like this, okay, or cavities. This is where their active site can be found, and this is only in small numbers of enzymes, right? Usually this active site can be found on the surface of most enzyme like this. Okay, now this active sites, right, has specific shape as it is formed from 3 to 12 side chains of amino acids in their primary structure. Okay, you can see from here, right, a zoom, closer look of this on the surface of this enzyme, this active site of this enzyme. This is what can be seen, right? The active site consists of 3 to 12 side chains of amino acids, right, in a perfect arrangement. Okay, so the arrangement of these side chains, okay, form the active sites. And then this active site has specific substrate to bind for each type of enzyme. Okay, shape of substrate must complement with the shape of the active site. Example, glucokinase is an enzyme that can only binds with glucose. So only glucokinase only able to binds and hydrolyze glucose. And one active site can bind with one or more substrates molecule. Okay. Now we have a diagram here, okay, that shows you the allosteric inhibition and allosteric activation. Okay. Now before we proceed with this, right, let's go to the second structure of an enzyme, which is allosteric sites. Okay. Every enzyme, right, not every enzyme, okay, some enzyme has allosteric site. Okay, and this allosteric site also known as the control site of an enzyme. Now, active site and allosteric site, they are two different locations on or in an enzyme. Right, every enzyme has active sites. Okay, let me rephrase that. Every enzyme has active site, while not all enzyme has allosteric sites. Okay, now this allosteric site, like I mentioned, like what I've mentioned earlier, right, it is known as the control site, right, of an enzyme. Why does it known as the control site? Because this allosteric site speeds up a reaction or slow down a reaction, or maybe stop a reaction, completely stop a reaction, right, when the allosteric site is specifically attach or bind to certain chemical. Okay, so meaning to say, if this enzyme, all right, okay, is already bind with a substrate, obviously this enzyme will hydrolyze that substrate or break down that substrate, betul kan? But in the process of breaking down the substrate, let's say suddenly an allosteric activator, okay, an allosteric activator, um, an allosteric inhibitor attached to the allosteric sites. What will happen? The substrate will not be broken down. Okay. Now, this allosteric site has specific shape as it is formed from 3 to 12 side chains of amino acids in their primary structure. So allosteric site is very much similar with the active site. Now, example, okay, phosphorylase action is activated by adenosine monophosphates, okay, but inhibited by adenosine triphosphate when adenosine triphosphate bound to the allosteric sites. So, meaning to say, okay, there are two modes of action for these allosteric sites, either allosteric inhibition or allosteric activation. Okay, now in allosteric inhibition, okay, when certain chemical attach with the allosteric sites, that particular enzyme will not, all right, will not break down the substrate that is already attached to the active site of that enzyme, right? Because the binding of the chemical molecule to the allosteric site of that enzyme will cause the active site of the enzyme to be distorted. Distorted maksudnya dia punya configuration itu bertukar. Right? Kamu boleh tengok dalam dua gambar raja ini, yang ini dia punya active site a bit wider, yang sini dia jadi narrower. So substrate dia boleh bind. Okay? Ataupun kalau substrate sudah bind, substrate itu akan terkeluar. Okay? Now, like one example here is the phosphorylase action, or action of phosphorylase. Okay? di mana phosphorylase dia tidak boleh apa ini phosphorylase ni dia tidak boleh jalankan fungsi enzim dia kalau allosteric site phosphorylase ini bound or bind dengan ATP. Okay, ATP. Okay, another example is allosteric activation. Okay, meaning to say, right, the active site of the enzyme is slightly distorted. So when the active site of the enzyme is slightly distorted, substrate cannot bind properly or cannot bind at all. Okay, so the allosteric sites of that enzyme, okay, needs to bind with a chemical molecule, right? Or to be more specific, the allosteric site of the enzyme needs to be bind with an allosteric activator. So when an allosteric activator bind to the when an allosteric activator bind to the allosteric sites, okay, allosteric site of that enzyme, what happens? The active site of that enzyme will change its configuration to be complement with the substrate. So this enables the substrate to bind with the active site of that enzyme. Alright, enzyme mode of action. There are 5 ways. Okay, there are 5 ways. So, first, enzyme lowers the activation energy of a reaction by forming an enzyme substrate complex. So, enzyme, we add with substrate, okay, an enzyme substrate complex will be formed, right? And eventually, after the substrate is already broken down by that enzyme, in that enzyme substrate complex, right, the outcome will be the enzyme itself and the product, okay, the substrate. So what happens? Enzyme lowers the activation energy, and you can refer here, right? A reaction that is uncatalyzed with enzyme, right, this is the activation energy that is needed, which is quite high, right? You see, free energy against rate of reaction. This is the uh, activation energy needed, okay, for any reaction that is not catalyzed by enzyme. But if that substrate is being added with enzyme, okay, this is the uh, activation energy needed to catalyze that reaction. Okay, sangat rendah. Okay, I think you have seen this graph so many times already. Okay, obviously an enzyme will lower the activation energy of any reaction that is being catalyzed by that particular enzyme, right? So what is activation energy? Is the minimal energy required for a reactant or substrate to start a reaction. Okay, now example, catalyzes the breakdown of hydrogen peroxide to form water, oxygen, and energy. So without catalyze, okay, higher activation energy is needed to break down hydrogen peroxide to water, oxygen, and energy. Okay, now second, enzyme weakens the bonds within a substrate, okay, causing the substrate to break and become smaller product. This enzyme carries out degradation of substrate. We nampak the degradation here, how this enzyme how the substrate bind with the enzyme to form the enzyme substrate complex, and then this enzyme in the enzyme substrate complex will break down the substrate into smaller products. And then the enzyme can be reused for the same reaction. Now, enzyme, okay, the third characteristic of enzyme brings two substrates molecules close together, how these two substrate molecules bind with the active site of this enzyme, right, to initiate a chemical reaction. So what happens? The enzyme in the enzyme substrate complex will break certain bonds in the substrate and joins the substrate to become a larger product. This enzyme carries out synthesis of product. So enzymes carries out degradation of substrates. Enzyme also carries out synthesis of product. Enzyme provides a template that orientates two substrate molecules facing each other to start a reaction. Okay, so meaning to say, when these two substrates bind with the active site of the enzyme will orientate the two substrate molecules to facing each other. Okay, and this starts a reaction. And then enzyme provides special microenvironment that favors the reaction to take place. Yes, inilah apa ini microenvironment tu, which is the active sites of the enzyme. Okay, because the active site of the enzyme favors for a reaction to take place. So this is due to the side chains of the amino acids that are able to donate or receive hydrogen ions from the substrates. Okay, now just a recap, okay, on some of this terminology, catabolism and anabolism. Okay, so we are done on the enzyme, all right, on the structure of the enzyme. So before we close our discussion, let's do some recap here. Okay, on what you have learned in your secondary education. Catabolism is a chemical reaction that results in the breakdown of more complex organic molecules into simpler substances. Okay, so simply to say, catabolism is a process where substrate or a molecule is broken down. Okay, and catabolism releases energy that is used to drive the chemical reactions. Now, on the contrary, with catabolism is anabolism. Anabolism is the chemical reactions in which simpler substances are combined to form more complex molecules. Okay, and anabolism require energy. Okay, the energy is needed to build the new molecules. Okay, or to store the energy itself. Now, the energy of catabolic reaction is used to drive anabolic reactions. Betul kan? Okay, and then the energy for chemical reactions is stored in the ATP. Okay, so that's it for this morning. All right, thank you. See you in the next video.